Welcome to Lecture 9 of BIOT 643. This week we'll be discussing the applications of genetically modified organisms. Over the course of this lecture we'll be presenting topics on bacteria, the protein production in bacteria, plants, the development of pest resistance, disease resistance, and increasing crop yields and nutrition. And finally, we'll be discussing these methods as they apply to animals, the methods, the production of useful compounds, and other applications. First, we'll start with bacteria. Bacteria are often used for protein production because they are easily grown and their simple genetics allow easy modification of their genome. Insertion of useful foreign genes into bacteria can allow the easy production of large amounts of protein in its heterologous system. The first such application was in 1978 when the gene encoding the human peptide hormone insulin was produced in E. coli. In 1982, this biosynthetic insulin was approved for use in humans by the FDA. Here, we show an example of insulin production in bacteria, with the question of how do they make insulin from recombinant DNA? And this shows you the overall process. Initially, you isolate the human insulin gene, the DNA. This is incorporated into a plasmid or a loop of bacterial DNA. As you'll recall from our earlier lectures, this involves using the restriction endonucleases to cleave and make sticky ends so that the human insulin gene can be ligated into the plasmid. This gene is then inserted into the plasmid and ligated. This now recombinant DNA, this plasmid which contains the human insulin gene, is then inserted or transfected into the recombinant bacterium. This now recombinant bacterium is then grown in large scale in a fermentation tank. And from this fermentation tank, you isolate the insulin which is produced from this recombinant bacterium and released into the culture media. The insulin is harvested. The insulin is then purified. And this purified recombinant insulin is then now marketed for treating humans. Previously, insulin was isolated from the pancreas of pigs. However, this approach was costly, it generated low yields, and had the potential for adverse reactions. The recombinant production of human insulin has many advantages. There's the cost savings. There are far greater yields, a higher purity. There's the decreased incidence of allergic reactions. And they eliminated the need to euthanize pigs to harvest their pancreas for the production of insulin. And there are a number of protein products which have been produced in bacteria. Here are just a few of the examples: alpha amylase, biofuels, hemophilia clotting factors, human growth hormone, insulin, vaccines. Now let's talk about plants. Many plant species have been genetically modified. And here in this table we show just a very short list of all of the plants that have commercial uses that have been genetically transformed. There are many reasons for creating genetically modified plants. to increase their disease resistance, to increase their resistance to pests, and to increase crop yields. And in 2014, a meta-analysis studied the agricultural and environmental costs and benefits of adopting genetically modified crops. These results include a 37% decrease in the use of chemical pesticides. There was a 22% increase in the crop yields. And overall, this resulted in a 68% increase in profits for the farmers. So what are some of the methods used for genetically modifying plants? Plant transfection is typically performed using the soil-dwelling bacterium, the Agrobacterium tumefaciens. The Agrobacterium tumefaciens is the plant pathogen responsible for the crown gall disease, in which large tumors form at the base of the plant. The bacterium transfers part of its genome into the plant genome. Here, on the right, we show you a diagram of those events. In this case, the TI plasmid, and the TI stands for tumor inducing. This plasmid is then injects the tDNA, that's transferred DNA, region of the TI plasmid into the plant cells. The tDNA segment integrates into the plant cell's chromosome, becoming a stable part of the genome. And again, here we show a diagram of some of those events showing the tDNA region. By replacing the native genes between the left and right borders, the TI plasmid can be used as a vector to transfect desired genes into plants. And again, here we show this tDNA region, and we show the left border and the right border into the area, which is the area where the desired genes can be transfected. This is achieved by the same process. You use an endonuclease to remove or open the portion of it. This leaves sticky ends where you can now transfect with a plant selectable marker gene and your target gene. And again, as we said, that's inserted between the left and the right borders of this TI plasmid. And in this case, we have a selectable marker gene for both the E. coli and the Agrobacterium tumefaciens. By adding the E. coli origin of replication, this allows the easy genetic manipulation in E. coli and creates a shuttle vector. So one of the methods that can be used is a microparticle bombardment. This involves small, heavy metal particles, generally gold or tungsten, that are coated with plasmid DNA. These are propelled by high pressure gas, such as helium. The particles are fired into a plant tissue sample. The particles enter the plant cells and release the coated DNA, which then incorporates into the plant genome. Here, and again, this shows a diagram of how this works, we show the high pressured gas. In this case, we're showing helium, which is used to fire these particles, these heavy metal particles that are coated with plasmid DNA, into, in this case, we show the sterile leaf, and there the particles enter the plant cells and release their coated DNA. So how can this approach be utilized for pest resistance? Using genetic modification, one can introduce disease resistance to plants. By genetically modifying plant genomes to include genes that can help fight off pathogens, plants can be reprogrammed to be resistant to such agriculturally devastating diseases. Example genes include chitinase, an enzyme that breaks down chitin, an essential component of the fungal cell wall, or glucanase, an enzyme, again, that breaks down glucan, an essential component of the fungal cell wall. The creation of genetically modified, disease-resistant crops can prevent severe economic loss and malnutrition caused by insufficient food supply. So how can genetic modifications increase crop yields and nutrition? A 2009 study by EG Economics Limited found that in Mexico, yields increase with herbicide-tolerant soybeans by 9%. In Romania, yield increased with herbicide-tolerant soybeans an average of 31%. In the Philippines, the average yield increase of 15% was observed with herbicide-resistant corn. Also, they saw a 24% increase with insect-resistant corn. In Hawaii, a 40% increase was observed when one used a virus-resistant papaya. And in India, insect-resistant cotton has led to yield increases on average of more than 50%. So here are some examples of nutritionally enhanced plants. For example, canola can be genetically modified to increase the vitamin A, the linolenic acid, or the omega-6 fatty acid. It can also have added beta-carotene, which is a vitamin A precursor. For coffee, genetic modification can decrease the amount of caffeine. For corn, you can increase the amount of vitamin C and increase iron. And the tomatoes, you can increase beta-carotene and the lycopene and increase flavonoids or the antioxidants. And one of the things we'll cover in a little bit more detail is in rice, one can add the beta-carotene. On the right of this chart, it shows the genes or transgene products which have been added to these plants to enhance their nutrition. So what is the issue with vitamin A deficiency? It is a principal cause of preventable blindness in children. It increases the risk of infections from common childhood diseases. It is a serious public health concern in over half of the countries, especially in Africa and Southeast Asia. Vitamin A deficiency results in between 250 and 500,000 children becoming blind annually, and half of these will die within 12 months of losing their sight. Beta-carotene is an orange hydrocarbon that is a precursor to vitamin A. It is commonly found in carrots, sweet potatoes, and pumpkins. This is responsible for their orange colors. Beta-carotene is converted by an intestinal enzyme into vitamin A, and the dietary intake of beta-carotene is important to health, with intake insufficiency leading to a vitamin A deficiency. So one can use these biotechnology methods to create fortified or genetically enhanced rice. In 2000, the genes for two enzymes were integrated into the rice genome. Here we show the pathway which leads to the formation of alpha and beta carotene. You have the geranyl-gerane pyrophosphate, can ultimately be converted into lycopene, which is then enzymatically converted into alpha and beta carotene. However, you can produce or introduce into these plants the PSY gene from the daffodil. In this case, you'll be entering the phytoene synthetase, as well as the CRT1 gene from a bacterium. Here you're adding the carotene desatronase, which catalyzes two of the key steps for the conversion to lycopene. And the last step, the lycopene cyclase, is naturally produced within these wild-type rice. So by simply integrating two genes, one from a daffodil and one from a bacterium, you can now enhance the production of beta carotene within rice. The results in a complete biosynthetic pathway for beta carotene. So this produces the golden rice. Grains of the beta carotene fortified rice take on a golden orange color due to the high concentration of beta carotene. The golden rice has the potential to significantly reduce vitamin A deficiency associated blindness and death globally. In 2005, golden rice 2 was developed, which produces more than 20 times more beta carotene than the original version. So now we'll discuss genetic modification of animals. Recombinant DNA technology can be used to create transgenic animals. The advantages of generating transgenic animals include establishing animal models to study human diseases, studying mammalian gene expression and developmental issues, expressing foreign proteins in animals, the production of donor organs, creating disease-resistant livestock, improving milk quality, and animal production traits. So what are some of the methods used to develop transgenic animals? A transgenic animal is created either by introduction of the trans gene into the nucleus of the cell, or through somatic cell nuclear transfer, where the nucleus from a variety of cell types is transferred into and replaces the nucleus of the egg. In both these methods, a transgene can be introduced into the fertilized egg using a retroviral vector, or direct DNA microinjection, or the use of embryonic stem cells. Following the birth of the transgenic pups, they are typically screened by PCR or by Southern blot analysis. On this slide, we show a cartoon demonstrating the creation of a transgenic animal, in this case, a transgenic cow. You start with the gene of choice is manipulated and prepared in the laboratory. This transgene is micro-injected into the egg of the animal. This egg is then implanted into a surrogate. And over time, then the surrogate gives birth to your now transgenic calf, which now carries the gene that was created initially. So how can this genetic modification be used to produce useful compounds? Well, this is a process known as animal farming with a pH. For certain protein drugs that require complex modifications or are needed in large supply, production of transgenic animals seems most efficient. These farm animals become a production facility with many advantages. They are reproducible. There is flexible production capacity through the number of animals bred. It maintains its own fuel supply. The drug is delivered from the animal in a very convenient form, in its milk. Some human proteins have already been produced in the mammary glands of transgenic animals, and here is just a very short list, including such things as antithrombin 3, alpha 1 antitrypsin, factors 8 and 9, growth hormone, hemoglobin, insulin, insulin-like growth factor, interleukin 2, a variety of monoclonal antibodies, nerve growth factor, and tissue plasminogen activator, to name a few. So what are some of the other applications of genetic modifications of animals? They can serve as biosensors, thereby the detection of environmental pollutants. By placing the GFP gene downstream of a promoter induced by estrogenic chemicals, a strain of fish has been developed which detects natural and synthetic estrogen compounds in water. When these exposed to these compounds, these fish become fluorescent when positive. By placing genes under the regulation of different promoters, the detection input could be changed to any chemical for which there is a known promoter. In this case, instead of the estrogenic promoter, you can now add any promoter for which you have a known chemical. In this case, rather than detecting estrogenic compounds, you can detect anything: aluminum, arsenic, cadmium, chlorobenzenes, naphthalene, toluenes, zinc, etc. That concludes our lecture. Here is a list of the references used in the preparation of this lecture and may be helpful for your review.